In the beginning was the Word. The Word. In the beginning was the Word. The Word was with God, and the Word was God. Hello. Hello. In the beginning was the Word. Hey, Karis. Aren't you cold in shorts? Oh, you're in a skirt. Okay. Aren't you cold in a skirt? You've got a chance, I guess, of snow. 10% chance. It snows up there pretty often, so that doesn't surprise me too much. You know, I've never been there. I understand it's quite pretty. Mm. I love it when it tells me to start, reminds me to start Zoom when Zoom has already been started. Not a very smart program, is it? Morning, morning, morning. Hey, Allison, your lab book's up there. There she is. Hi, Ellen.
I'm good. I'm not sick.
Welcome back. We missed you. Okay, sports fans, let's go ahead and get started. So we are talking about post-parasite, post-pathogen interactions. We'll get today into virulence factors and those sorts of things, how bacteria colonize the body, what sort of tools they use to cause disease. They have to be able to colonize. To do that, they have to be able to attach, to invade, so on and so forth, avoid host immune processes, stuff like that. And we'll see how they do that. They get a little help from their friends, shall we say. Can you please put your mask on? Thank you. Oh, okay. You're drinking water. All right. Let's have a word of prayer. Father, thank you for this day. And we thank you, some of us have returned from quarantine. Lord, I think all of us are a bit suffering from a bit of COVID fatigue. I think we're tired of wearing masks. We're tired of having to be separate from everybody else, tired of being dragged apart by rules meant to keep us safe, but rules that just really seem to wear us out. Lord, we all need, I think, a new infusion of strength, a new infusion of patience, of tolerance. Give us that, Lord. Get us through to the end of the semester. Thank you, people that are coming off quarantine. We pray we would not have new people going into quarantine. Thank you for this day. And depending on how you feel, thank you for the snow if it appears. In Jesus' name, amen. Yeah, 10% chance of snow. So I don't know how you feel about snow, but you live in Michigan, it's inevitable, right? Very good. So we talked before about Koch's postulates. So Robert Koch, the German physician who first grew bacteria on a slice of potato, and then from a friend of his wife's, discovered agar because she was into Asian cooking. And we owe the use of agar in microbiology to Fanny Hess, who introduced Koch to agar. Koch designed so-called Koch's postulates. So these postulates simply dictate that what must be observed in order for you to conclude conclusively that a microorganism causes a disease. Now, you'll notice right away, this is taken from the slides that come from the book. You'll notice there's a pretty big typo right there. It should be of. Hello, hello, hello.
Oop, I can hear you now.
Yeah. It's shut off for some reason. I don't know why.
Oh, that's okay.
Well,
I just, all of a
that's,
sudden, I couldn't hear you.
well, it affected the recording, that means, which I don't know how much of the recording. It's bloody technology. You live or die by it. So,
I know it.
okay. Yeah.
My question, I'm a little bad before you three. I was trying to look this up, but I wasn't, like, a few of them I feel like could apply, so I'm not really sure, if these are right, and I couldn't find this one.
Aeruginosa, the best drugs for Pseudomonas aeruginosa are either, there's a vaporized, well, an aerosolized form of it for people who get Pseudomonas aeruginosa pneumonias, usually people with cystic fibrosis. It's called Caistin, so it's trianum would be one. The other, the mainstay for that is piperacillin tazobactam,
Okay.
which is, oh, what's the trade name of that? Piperacillin tazobactam. It's, can never remember that. I remember piperacillin tazobactam, but not.
So I wasn't sure because it said from the above table, so do we not have to take it from up to?
Yeah, well, none of those antibiotics work very well
Okay,
on Pseudomonas. Okay, let's see. Gentamicin should work on that one, but let's see. Oh boy. No, that's the, okay, there. There it is, Zosyn.
okay.
Piperacillin tazobactam, as you can see here, it comes in a generic now.
Gotcha.
That one you can only give intravenously.
Okay.
You can't give that by mouth.
Okay.
The other, piperacillin is not usually given. Say it's discontinued. It's usually given with tazobactam, which is a beta-lactamase inhibitor.
Okay.
The other drug that is given for Pseudomonas is this one right here, ceftazidime. That specializes for Pseudomonas. You can see that one's only available for injection. Gentamicin would be the other one. Again, it's a cephalosporin, excuse me, it's aminoglycoside, so you've got to be careful of kidney function and otic function. But as aminoglycosides go, gentamicin is the safest.
Okay. And then would the other ones be correct? Because I was just
Yeah, vancomycin, our staff was pretty resistant to vanco.
Yeah.
Which is kind of scary. Yeah, vanco is kind of the mainstay for staff nowadays. The other option would be linezolid or cefepime.
Okay.
Ceftaroline would be the other one.
Okay.
This one. Yeah. Teflaro.
Okay.
That one's a specialist for MRSA.
Okay. All right. I think those were my questions. Thank you.
Yeah, no problem. Okay, gang, let's go ahead and get back in the saddle here. All right, so as you can see in this sequence of slides, we have an initial attachment, we have an invasion. Notice, to invade, it has to degrade the junctional complexes that hold the cells together. Once it then gets into the deeper tissues, it needs to be able to deal with host defenses, both specific and non-specific. So we have a clot essentially that might form around invading organisms. In the case of some organisms, forming clots actually can protect them from damage, from being detected by the immune system. That is the rationale behind this thing you've been looking at this week in lab, coagulase. So Staphylococci like to invade the skin, and if they invade the skin, they can induce little blood clots around themselves, and then the immune system essentially comes sniffing around, and they say, blood clot, nothing to see here, let's move on. And then the organism will break out and then find their way into other places. This organism here, Streptococcus, actually has an enzyme to degrade clots, because sometimes clots can be used to wall off organisms to prevent them from invading further. Many organisms have proteases that can do this, and kinase is actually specific for degrading blood clots. Streptokinase is a product actually that's commercially available to degrade blood clots, and it's used as a clot buster. It's a little hard to control, so it's not used nearly as much as TPA, but it is used. It has its places to be used. So here's a list of some of the different types of toxins that you see in bacteria. So endotoxin refers to the gram-negative lipopolysaccharide. Gram-negative lipopolysaccharide can cause a very high fever. And it also will cause white blood cells to divide very robustly. And it's responsible for the very high fever you get in certain types of sepsis. So when you hear endotoxin, that is a bacterial lipopolysaccharide on the outer cell membrane of gram-negative bacteria. Exotoxins are secreted by bacteria, either gram-positive or bacteria. And they can either be enzymes or they can be toxins or what have you. Some of these toxins are toxic to white blood cells specifically, so they're called leukocidins. Leukocidins. And that is misspelled. That should be toxemia. The N shouldn't be there. Scratch that out for me, would you please? Toxemia means the organism has not invaded your body. The disease that you have is the result of the bacterial toxins. The best examples of these are food poisonings. Food poisonings. So staphylococcal food poisoning, for example. Staphylococcal food poisoning is marked by the fact that it comes on early and it leaves very quickly. So it's usually 1 to 6 hours after exposure. It usually lasts anywhere from 2 to 6 hours, and then it leaves you. Does the organism colonize your body? No. It's in your body, secretes toxins, passes through, does its thing, and then it's done. That's a toxemia. Some types of diseases are solely the result of toxins. They're not due to the organism colonizing your body. That's a toxemia. Intoxication refers to a condition that is the result of decreased. Good example here would be toxic shock syndrome. Toxic shock syndrome. So toxic shock toxin is released by some strains of Streptococcus pyogenes and Staphylococcus aureus. And these can have broad systemic functions, broad systemic consequences that will affect your whole body and will cause a sepsis-like condition where you get increased leakage through blood vessels, your blood pressure crashes, and so on and so forth. That's a toxemia. Is it because is it because the organism has invaded your body? No, because of the toxin. We talked about neurotoxins, sorry, enterotoxins. Enterotoxins affect the GI tract. They cause you to have increased secretion, or it causes the vomiting response. Neurotoxins affect nerves. They prevent nerves from working, or they may prevent nerves from relaxing. They affect nerves. They affect nerves specifically. Botulinum toxin, tetanus toxin. These are examples of neurotoxins. Toxoid. Toxoid. Toxoids are toxins that have been modified. They no longer cause you damage, but they are recognized by the immune system. So they give you resistance. They vaccinate you against that toxin. They vaccinate you against that toxin. Your tetanus booster is a toxoid. You gain a very robust immune response against the toxin. Now, if you're exposed to that toxin, it won't cause you that much damage because your immune response to it. Just some features about toxins. Some will produce fever, some won't. Some have broad effects on tissues, others are very specific. Some are very toxic in very small amounts. Some you need a lot of it in order for it to have a biological effect. Some are very stable. Some are heat stable. Some are heat labile. Some are acid stable. Some are acid labile. It depends. Mostly polypeptides, but not always. They are usually secreted. Usually secreted. Your immune system usually will recognize them and will mount a fairly robust immune response against them. And to convert them to toxoids, there's lots of ways to do that. Your book gives you some examples. I'll let you read about those. And here are more examples. Blah, 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 so on and so forth. Extra, read all about it. Go ahead and read about those. Okay. Endotoxin. Endotoxin tends to be sloughed while the cell grows very slowly, but when the cell dies, it's kind of released en masse. It can cause fever, and it can cause shock, and it can cause a condition called disseminated intravascular coagulation, or DIC, which we talked about last year in pathophys. Gram-positive organisms will secrete exotoxins. They don't have endotoxin. They will secrete exotoxins. This is a little deceiving because gram-negative bacteria can also secrete exotoxins. So that you might want to put a slash gram-negative as well, since they can also secrete exotoxins. Usually proteins, most time enzymes, not always. They will cause some sort of specialized tissue damage. They're almost always secreted. Almost always secreted. There are a few toxins that are the result of sporulation. And so they're part of the spore. They're called a parasporal body. So if you ingest spores, you'll have the toxin essentially as a result of sporulation. Others, like for example, anthrax, you ingest the spores, the cells germinate, then they secrete the toxin. Others, they're associated with spores, but mostly they're secreted. Now, hemolysins can be detected on a blood agar plate. And if you get this, boy, that's hard to see. Let's try something. You see that better? Or no? It's supposed to be green. Can you see the green? Is that better or not really? You can see it? That greening reaction is a result of so-called alpha hemolysis. Alpha hemolysis. Now, why does it turn green? It turns green because the damage to the cells converts the hemoglobin to methemoglobin. And methemoglobin has green color. So that greening is the result of alpha hemolysis. Also, the damage of the red blood cells, if you hold the plate up to the light, seeing this on your blood plates, the plate looks as though where the bacterial cells are, the blood cells around there are lighter colored than the ones that don't have any bacterial cells on them because the cells have been damaged. That's alpha hemolysis. Beta hemolysis, of complete destruction of the blood cells, you hold the light, you can see straight through it. That's beta hemolysis. The cells aren't just damaged, they've been lysed. They've been lysed. And if you see beta hemolysis on a blood plate of an isolate from a patient, that should give you pause. How about viruses? Viruses are really going to depend on the virus and the type of life cycle that it leads. One of the first things you see, at least with DNA viruses, the cells will swell up, particularly the nucleus. So the cells, they swell. The nucleus may also swell. Some viruses will give you so-called productive infection, where the cells bud virus at a certain rate. They don't really kill the cell. Others are very cytolytic, and they kill the cell. So a good one is poliovirus. If you grow poliovirus in a lab, the cells it affects, boom, blows them up. It's very, very cytolytic. Others, you get little inclusion bodies in them with rabies virus. And then others, you get clumping, actually, of blood cells together. That's the case with influenza. And also, Similiki forest virus will do that. And so will para-influenza viruses, too. Here is an example of cells that have been grown on a virus. Notice some of these cells are kind of trapezoidal looking. Others are more roundish. These cells are infected by a virus. They typically detach and they round up. So they have a different look from the other cells. They behave differently too. Now, some RNA viruses, because your RNA polymerases are so air prone, they will tend to make particles that are defective. And those defective particles might infect other cells, but they will produce a so-called abortive infection. Abortive infection. They'll start to infect and then it's kind of nothing doing. Latent infections. Some viruses can either kind of hide inside the cell, infect, or they can go past like $200 and infect the cell, blow it up, make new viruses, so on and so forth. If they do that, they risk the wrath of the immune system. What if you hide inside the cell and wait for the immune system to kind of take a dip and then come out? Well, that's latency, and that is a feature of various types of herpes viruses. Then there are some viruses that cause so-called persistent infections. You have this low-grade infection that does not cause a robust immune response. Because of this low-grade infection, doesn't hurt the host very much, but you're kind of constantly shedding virus. And there are a couple of viruses essentially that can do that. But that's persistent infections. Algae, yeah, there aren't a lot of algae that cause disease. There's one, and it's only really infects people who are immunosuppressed. These types of eukaryotic pathogens, they will tend to cause pretty robust inflammation. That robust inflammation is very hard to miss. So these infections usually will be on the fulminant side. The exception comes with some of your helminth infections in the GI tract. Because humans have existed with helminth infections for so long, some of these essentially can cause low-grade infections that really you don't even know you have them. But many of these others, if you get infected by them, your response that you get is usually quite robust. What's the difference between a symptom and a sign? There you go. There you go. Say it again. Symptom, that's what the patient tells you. I feel X. I feel X. I feel tired. I feel lousy. I have a fever. I have a sore throat. It hurts when I bend over. Hurts when I blink. My neck is stiff. Those are symptoms. Signs: temperature, blood pressure, heart rate, flushing. Pull the eyelid down. Look at the inside of the eyelid. Is it red? So on and so forth. That's what you see, what you detect. Syndrome. Syndrome. Syndrome is a group of symptoms that consistently occur and are associated with a specific condition. It's a group of symptoms that consistently occur and are associated with a particular condition. That's a syndrome. Sequelae. Sequelae. These occur after the disease has resolved. They may occur soon after. They may occur years after. sequelae. It might be because of the immune response to the disease, or it might be because the organism hasn't been completely cleared from your body. Acute disease. Acute disease. You get it, you get a sign of disease very quickly. Acute disease. Chronic disease, you've had it for a while. And the cutoff usually is 6 months, typically. Subacute disease. That's kind of that in between no man's land. Between right away, takes a week, takes 2 weeks, so on and so forth. That's subacute. Latent disease. That's an infection that's hidden or dormant, as opposed to an active infection, where the bacteria, virus, whatever is actively dividing and causing symptoms. Latent infections are usually static. Active infections are usually dynamic. Focal infection. Focal infection. You have an infection localized around a certain area. From there, it can spread to the rest of the body. So the focus of infection is the abscess tooth, the thorn you got while gardening, the catheter, the implant, the, whatever. That's a focal infection. Infection localized at a region from which it can spread to the rest of the body. Local infection stays where it was introduced. Stays where it was introduced. And then systemic infection, it's spread throughout the whole body. Or it includes multiple systems, multiple organ systems. So bacteremia, septicemia, viremia, those fall under the general condition of sepsis. A bacteremia is sepsis caused by bacteria. Bacteria have colonized the blood. A viremia, viruses have colonized the blood. Viremia is actually part of the infective cycle of some viruses. A fungemia would be a septicemia caused by fungal organisms. And then a parasitemia would be eukaryotic colonized and affected the blood as well. All of those are examples of a septicemia, where the blood is colonized by an infectious agent. Sapremia. Sapremia. That is a toxic state resulting from the presence of usually a putrefaction of an organ or tissue or something like that. So gangrene or some of the part of the body that is putrefying because of infection, that is a sapremia. The toxins that have built up in that part of the body are starting to now have systemic effects. That's a sapremia. Primary infection, that's your initial infection. Secondary infection, that follows up the primary infection. So for example, measles. Measles will tend to suppress the immune system after you get it, usually for about 6 months. So it's not uncommon for kids who get measles to get really severe secondary infections. That's another reason to vaccinate your kids. Oh, measles is just a childhood infection. People get it, they get over it. Well, what about secondary infection they might get from it? Super infection. Now you're infected with 2 infectious agents, one on top of the other. A super infection. Mixed infection. You're infected with more than one organism. So a lot of your peritonitis infections are like this. A lot of tooth abscesses are mixed infections. Inapparent infection. You have a definite infection, but there's no signs or symptoms. Example, poliovirus. 90% of people who get poliovirus don't know they have it. They have an inapparent infection. Now, why is this medically significant? Because people with poliovirus are still shedding it into the water system. This is a case with COVID-19, is it not? Because 25 to 50%, maybe more, of all people who get COVID-19 don't seem to show any signs or symptoms. Can they still pass the virus? Yes, they can. So that is an example of an inapparent infection. Okay, and here's some definitions. Compare those essentially to the ones I gave you. And this is the trajectory, if you will, of an infection. So the period of time between when you are infected and when your first symptoms appear, that's called the incubation period. The incubation periods vary wildly. For some, it's hours. For some, it's days. For some, it's weeks. When you first start to show symptoms, you usually have a period where you show very general symptoms. This is called the prodrome. In that period during which you have the prodrome, that is a prodromal period. Prodromal periods vary wildly. Then you have the invasive phase. Now you start to see the most severe signs and symptoms, usually signs and symptoms associated with that disease. In the prodrome, the symptoms tend to be pretty general, pretty vague. In the invasive phase, you see things more specific to that disease. You then have the acme. That's a point at which the disease is at its worst. The immune system is fighting the disease. Your fever is your highest, so on and so forth. Then you have the decline phase where the signs and symptoms start to decline, and the number of organisms in your body usually start to decline as well. And then you will go through the convalescent period where you're getting back to more 100%, that sort of thing. Convalescent phase can vary from disease to disease to person to person. How long the patient is infectious, that also varies from person to person as well. When they put this on a slide, it's the size of a postage stamp. And it's very hard to read, but these are examples of things you get in the prodromal period: headaches, generalized swelling. That would be an example of prodromal sorts of signs and symptoms. The invasive phase, we have cough, sore throat, fever, swollen lymph nodes, skin rashes, nasal congestion, pain at very specific sites, nausea, vomiting, diarrhea, and blah, 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 so on and so forth. During the convalescent period, the patient regains strength, but again, how long it takes for the patient is non-infectious, that's going to vary. Okay, so factors that influence infectious disease. This is something you'll talk more about in your community health class, but healthcare availability is a biggie. Also, what types of healthcare are available. It also depends on the adaptability of itself. It's very clear, for example, that when we read about syphilis, when explorers brought syphilis back from the New World, syphilis was a very fast-moving, very disfiguring disease. And because it disfigured people, you knew right away that someone had it. And the chances of somebody with disfiguring syphilis of passing it on was not as high. Now syphilis is a much slower moving, more insidious disease. So that's an example of a disease adapting to change. Also, migration patterns can also affect disease distribution, what have you. So can this phenomenon, we'll talk more about it, herd immunity. Herd immunity, that's a percentage of the population that are resistant to this particular agent. And then immigration, national travel, so on and so forth, that's a whole big kettle of fish there. And this just shows essentially mortality rates with infectious diseases. You can see with the development of penicillin, Salk vaccine, vaccination assistance acts, so on and so forth. Infectious disease now has become a pretty minor killer. Most of it now is heart disease, cancer, that sort of thing. That's why we're living longer. Infectious disease, as you can see, is that's good news, but infectious disease still manages to kill a lot of people each year. So there you are. All righty. I am going to, please. I'm going to stop right there, and then we will get into the next chapter. And I think the next chapter is epidemiology. We'll whiz through that, and then we'll get into the immune system. And we'll talk about that, then we'll get into specific diseases. To do the antibiotics. So E, that's erythromycin. P is penicillin G. VA is vancomycin. TE is tetracycline. STX is trimethoprim-methylmethoxazole. CRO is ceftriaxone. That's what's the name of the antibiotic
Is it
based on the
okay
code.
if I don't write down to the name? They don't know how to write those down. Or
You don't know how to write those down? Penicillin?
Oh, I can do it.
Penicillin G? Okay, I'll see you when I see you.